Okay, so today we're diving into something pretty fundamental that's changing in science. AI isn't just, summarizing things anymore. It's actually getting involved in the discovery itself.
That's exactly right. We're seeing AI generate hypotheses, run complex analyses, stuff that would normally take researchers, well, ages.
And that's what this deep dive is about. We've got sources on two systems really pushing the boundaries here.
Mm-hmm. First, there's KRONOS. Think of it as a specialist foundation model for biological imaging. Really complex stuff.
Okay, KRONOS for imaging. And the second,
That's KOSMOS. This one's more like an autonomous AI scientist. It can run discovery projects, basically on its own.
autonomous. Wow.
So yeah, our mission today is to unpack how KRONOS handles that messy imaging data, gets it clean and usable efficiently.
And then how Cosmos takes that kind of efficiency to the next level, actually starting to generate new scientific ideas.
Exactly. It's all about how these tools can help speed up knowledge acquisition for, well, for you listening.
Let's start with Kronos then. It's tackling spatial proteomics imaging, right? That sounds complicated.
It is. You're mapping thousands of proteins, often in single cells, within tissues. You get these incredibly rich images.
But the richness is also the challenge.
Precisely. The traditional way to analyze these images involves segmentation, meticulously drawing boundaries around every single cell.
I can see how that'd be tough.
Yeah, especially in complex tissues like tumors or where there's damage. The cells are crowded, irregular. Segmentation often just breaks down. It fails.
So your analysis downstream is compromised from the start.
Exactly. Garbage in, garbage out, basically. Or you just can't analyze it at all. It's a huge bottleneck.
Okay, so Kronos had to learn a different way. How did it manage that? What was the training like?
The scale is kind of mind-blowing. It was trained on a dataset called SPM47M.
SPM47M, what's the number mean?
47 million. Over 47 million image patches.
Whoa, okay, 47 million, that's a lot of images.
It really is. And crucially, it wasn't just volume, it was the diversity.
Ah, right. What kind of diversity are we talking about?
Well, it covered 175 different protein markers, 16 different tissue types, brain, kidney, tumors, healthy tissue,
16 tissue types.
and data from 8 different fluorescence imaging platforms. So different machines, different setups.
Okay, so it learned the general patterns of cells and tissues, not just specific quirks of one data set.
That's the key. It developed these really robust general-purpose representations. It learns what a T-cell or tumor microenvironment fundamentally looks like, regardless of the specifics of how it was imaged.
That generalizability must be huge for labs. Annotation is so expensive and time-consuming.
Absolutely. And this is where KRONOS really shines its data efficiency or label efficiency.
How well does it actually perform compared to other models?
Significantly better, especially on tough tasks.
Yeah.
Take cell phenotyping in, say, classical Hodgkin's lymphoma, CHL, really complex visually.
Okay.
KRONOS hit a balanced accuracy of 0.7358.
0.73. And what's the benchmark? How good is that?
Well, compare that to something like DynoV2, which is a very strong general vision model. It only managed 0.6210 on the same data.
Okay, so a noticeable jump. Kronos' specialization really pays off.
Definitely. But it's not just about being better with lots of labels. It's also really good with few labels.
Ah, the efficiency part.
Right. It maintains high performance even with minimal supervision. Like, it did much better than baselines when trained with only 100 labeled examples per cell type.
Only 100. That's tiny for deep learning.
It is. And it even enables really effective one-shot annotation scenarios. You give it one example, and it can generalize surprisingly well. That changes the game for research, where getting labels is the bottleneck.
That leads to this different way of thinking about analysis. You mentioned segmentation-free. How does that work? No more drawing cell boundaries.
Exactly. Forget drawing perfect lines around every cell. KRONOS looks at small image patches instead. Yeah, small squares of the image. It analyzes the texture, the patterns, the neighborhood within that patch, without needing to know precisely where cell A stops and cell B starts.
So it's more about recognizing the local environment, like the forest, not just counting trees.
That's a great analogy, yes.
Yeah.
It allows analysis across scales. You can understand individual cell types present, the characteristics of the immediate microenvironment, and even the larger tissue architecture, all from these patch-level features.
Less brittle than segmentation, sounds like.
Much less brittle. It handles the messiness of real biological data much better.
And we see that in practice, like identifying tumor regions.
Yep. In prostate cancer data, it was excellent at delineating tumor boundaries. Achieved an AUC area under the curve of 0.91.
And how does that compare?
Well, a baseline using just marker expression levels, which is closer to what a pathologist might assess visually, only got 0.68.
Big difference. It's seeing more subtle patterns.
Exactly. And it also helps with just basic data quality. It's really good at spotting artifacts. Blurred regions, areas where the signal is too bright, saturated, common problems in fluorescence microscopy.
Oh, right. Experimental noise.
KRONOS detected these with 0.98 mean precision. It can automatically flag or even filter out the bad data before it messes up the analysis.
So KRONOS acts like an expert eye, cleaning, standardizing, and interpreting these complex images without getting bogged down in segmentation.
And that leads to this idea of it being a kind of spatial proteomics image reverse search engine.
Reverse search engine meaning?
Meaning a scientist can take a specific pattern they see, maybe a weird cell cluster or a particular tissue structure, and ask KRONOS, find me other examples like this across all our data sets.
Even if those data sets came from different labs, different machines.
Exactly. It bridges that gap, allowing large-scale comparison and pattern discovery across previously incompatible data. A huge win for collaboration.
Okay, so KRONOS provides this superpowered standardized input, which leads us perfectly to KOSMOS. If KRONOS handles the data quality, KOSMOS handles the discovery using that data.
That's the idea. KOSMOS is pitched as an AI scientist for autonomous discovery.
Autonomous. So you give it a goal and data, and it just goes.
Pretty much. It runs these iterative cycles. It analyzes the data, searches the relevant scientific literature, generates hypotheses based on both, then plans the next round of analysis. It keeps going, refining its understanding.
How do you make sure its reasoning is, well, reasonable, not just making stuff up? Traceability is key, right?
Absolutely. It uses a structured approach. There are two core AI agents.
Yeah.
One for data analysis, one for literature searching. They're coordinated by something called a world model.
World model, like a shared brain.
Sort of. Think of it as a central structured database. Every finding, every piece of evidence from the data, every relevant statement pulled from a paper, it all gets logged and linked in this world model.
Ah, so you can follow the chain of logic.
Exactly. The final report KOSMOS generates has every conclusion explicitly tied back to the supporting evidence in the world model. No black boxes.
And the scale it operates at is intense.
Oh, yeah. An average run might involve executing, say, 42,000 lines of code for analysis.
42,000.
And reading and synthesizing info from maybe 1,500 full scientific papers.
1,500 papers in one run.
Yep. Compared to earlier systems, it's generating almost 10 times more analysis code. It's compressing maybe months of human expert work into hours.
Okay, let's talk about the results. Did it actually discover anything interesting? What are the aha moments?
Definitely. First, there was a really important validation case, reproducing a known finding.
Okay, what was that?
It involved mouse brain metabolomics data. The context was studying how cooling the brain after neural activation protects its cerebroprotection. Human researchers had already figured out a key mechanism and published it.
And KOSMOS.
Working completely independently, just given the data and the objective, KOSMOS arrived at the exact same mechanistic conclusion.
Which was?
That the nucleotide salvage pathway gets activated. Basically, the brain cells start recycling nucleotide components to save energy and keep essential pools topped up during stress.
Okay, it found the same pathway, but how close were the details?
Almost identical. They compared the quantitative results, the fold changes in key metabolites, between KOSMOS's analysis and the human analysis. The correlation, the R-squared value, was 0.998.
0.998. That's basically perfect agreement.
It is. It showed KOSMOS wasn't just guessing. It could quantitatively replicate complex biological findings with extremely high fidelity.
Impressive validation. But did it go beyond replicating? Did it innovate? You mentioned something about Alzheimer's disease methods.
Ah, yes. This is where it got really interesting. The task was to understand the sequence of events in Alzheimer's progression, particularly involving tau protein buildup and damage to the extracellular matrix, the ECM, which is like the structural scaffold of the brain.
Okay, timing is crucial there.
Right. And the typical human approach might be to compare early-stage samples versus late-stage samples.
Makes sense.
But KOSMOS reasoned that this kind of binning, putting things into discrete boxes, wasn't precise enough to really nail down the exact timing relationship between tau and ECM failure.
Wait, so the AI critiqued the standard analysis method?
It did. And then it proposed a new one.
It invented a method.
Essentially, yes. It suggested using segmented regression analysis applied along a pseudotime axis.
Okay, pseudotime axis. Let's break that down.
Think of pseudotime as a calculated timeline of biological progression. You mathematically order your samples, like cells or tissue regions,
Right.
based on how far along a process they seem to be, even if they were collected at the same actual time point.
Right, ordering by biological stage, not just clock time.
Exactly. Cosmos used this pseudotime axis in the segmented regression to find a specific breakpoint, a point in the progression where the ECM suddenly starts to fail more rapidly.
That sounds like a much more precise way to pinpoint events. What did it find?
It found the breakpoint for ECM failure occurred at 0.58 units along this pseudotime trajectory. And importantly, this breakpoint was after the point where tau accumulation really took off.
So the ECM failure seems to happen downstream of the tau problem.
That's the conclusion. It established a specific biologically plausible sequence, tau pathology accelerates, then the structural matrix fails. And it did this using a method it effectively devised for the problem.
That's genuinely innovative. Okay, what about uncovering something completely new, a mechanism nobody had proposed?
Yes, the third example is really cool. This involved neuronal aging. Comparing vulnerable neurons in the entorhinal cortex, the ENT, an area hit early in Alzheimer's, with more resilient neurons from the main cortex, the CTX.
Okay, vulnerable versus resilient cells during aging. What did Cosmos spot?
It found a significant widespread downregulation, a decrease of specific proteins called P4-ATPase flippases, specifically in those aging vulnerable ENT neurons.
Flippases. What do they...
They're like molecular gatekeepers for the cell membrane. They maintain the balance, the asymmetry, of lipid molecules between the inner and outer layers of the membrane.
Okay, maintain balance. So what happens if they're downregulated, if they stop working well?
The membrane loses that crucial asymmetry. And one specific molecule, phosphatidylserine, which is normally kept hidden on the inside surface, starts appearing on the outside.
And phosphatidylserine on the outside means...
It's a well-known signal. Basically, it's an eat me signal for the immune system. And Cosmos didn't just stop there. Its literature search agent simultaneously found evidence that nearby immune cells, the microglia, were showing an upregulation of genes involved in sensing phosphatidylserine and triggering phagocytosis cellular eating.
So it connected the dots. The vulnerable neurons lose their flippases, display the eat me signal.
And the local immune cells get primed to recognize that signal and potentially remove those neurons.
Wow, that's a complete novel mechanistic hypothesis for why those specific neurons might be selectively vulnerable in aging and Alzheimer's, proposed entirely by the AI.
Exactly. Linking the molecular detail of flippase function directly to a potential mechanism of targeted cell removal. Experts rated this finding as having moderate to complete novelty.
Okay, let's synthesize this. KRONOS gives us incredibly high-quality, analysis-ready spatial data, solving the input problem, especially the labeling bottleneck.
Right. Robust, quality-controlled, label-efficient imaging analysis.
And then Cosmos takes data like that and runs these massive multi-pronged discovery campaigns, analyzing data, reading literature, generating and testing hypotheses.
Autonomously. It perfects the application of the data, churning out these deep, traceable scientific reports proposing novel mechanisms and findings.
So for the researcher listening, the big advantage is what's less grunt work?
Definitely less grunt work. It takes away the immense cost and time of manual annotation and the initial broad exploration phase of analysis. Human experts can then focus their efforts on the most promising, the most novel hypotheses flagged by the AI.
Focusing on the high-level validation and follow-up.
Precisely. And these aren't just theoretical findings. Cosmos has pointed to potentially actionable things, like identifying the SOD2 protein as potentially reducing heart fibrosis, suggesting a drug target. The flippase mechanism in AD could also open new therapeutic avenues.
So we've seen AI transforming how we handle complex imaging with KRONOS and how we conduct the research process itself with Cosmos.
It's a powerful combination.
Yeah.
But I want to leave you with one final thought, circling back to that Alzheimer's discovery by Cosmos.
The invented analysis method.
Exactly. The segmented regression on pseudotime. Cosmos didn't just apply existing tools better or faster. It recognized a limitation in the standard methodology and created a new quantitative approach specifically for that biological question.
The tool started improving the rules of the game.
Right. That's a profound shift. It's not just replicating human science anymore. It's adding new ways of reasoning, new analytical tools to the scientific toolkit. What happens when the AI systems we build start routinely inventing novel quantitative methods that we as humans wouldn't have thought of? The potential for accelerating discovery feels almost exponential. It really pushes us to think about the future depth of reasoning AI can bring.